everybody. Thanks a lot first, all the organizer for giving me the opportunity to give this talk here. So I'm Berenice Batut. I'm a postdoc at the University of Freiburg in Germany. And today I will give a talk for the holistic bioinformatic approach used in Microbiome Research conference. And I will talk about my project ASaiM and how it started from a local project to a global effort to support microbiome data analysis. And the story will start in 2015. So at this time, I just finished my PhD in evolution, mostly studying evolution within two different computational biology lab. And before I did a master in bioinformatics and computer science. So I had really a background in bioinformatics, but mostly on the computer science side. And then after my PhD in 2015, I joined a wet lab as the only bioinformatician in this lab. So I felt a bit like this mouse there, a bit like the computational mouse trapped in somewhere where I don't know anything about. And was, okay, it was quite interesting, that feeling there. And the lab there were mostly working on gut microbiota. They were studying how the gut microbiota interact with the host, and mostly focusing on human, but also on the crows. And they were doing some different type of analysis, so amplicon data analysis, so focusing on the 16S, but also metatranscriptomic and metagenomics data analysis. So they extracted the DNA, the RNA, and they sent everything to sequence, mostly using Illumina. There were a bit of 454 at this time also still. And the work on the bioinformatician, and it was mostly the idea was to be on my shoulder, was to extract the useful information from these sequences. So which organisms could we find in this gut microbiota, and what are the functions they are doing and how they are doing that. But it was not so my job there was not just to be a service somewhere and doing all the analysis for them. The context of my project was mostly they wanted to compare their data to available data on public databases. So at this time, there were different public data repository available with some metagenomics data. So for example, the European Nucleotide Archive, ENA, the NCBI, the DNA Data Bank of Japan, the DDBJ. And if we look, so we are back in 2015. If we looked for human gut metagenomes on ENA, and this time there were around a bit more than 200 studies, a bit more than 1,500 available data set for that. So not as much as we can find nowadays there. But so the idea of the group there, they wanted to be able to, once they have the data, to compare if the species that they have, but also the genes that they could find or identify on their samples, are they similar to things we could find already in databases. But the thing is, the data there were quite dispersed and really not comparable. I mean, you could find the data everywhere. You needed still to process them in a comparable way to make sure that we could compare between across different studies or across different samples also. So the aim of my project back then was to really collect gut microbiota data sets in different public databases, especially the one I mentioned, and analyze them given a standard workflow, which was mostly, and we were focusing mostly then on metagenomics and metagenomics data. So doing a quality control, taxonomic analysis to identify the community structure for the different species and different taxonomic levels we could find and with their abundances, and the functional analysis, so identify the genes or gene families, but also different pathways. at this time, there were different existing tools available. So the main one that you may have heard about are ASaiM and Mother. So ASaiM and Mother are two, the main tool suites to analyze amplicon data, and especially 16S data. They were quite popular at this time, and they are still quite popular. There were also MGRAST, mostly focusing on metagenomics data, where you could submit your data there and they were doing some analysis for you there. MetaMOS was also a pipeline for analyzing metagenomics data. But none of them were really, so we could get data from the different, from these different type, using these different platforms, but they were not really using a gut microbiota specific database, so we couldn't identify specific things we are aware about in gut microbiota. We couldn't really, so ASaiM Mother were focusing on 16S, but we were targeting mostly metagenomics data. So we were losing a bit of information if we are focusing only on the 16S there. And I didn't really mention until now, but the idea was also that the people in the lab there could be able to do their own analysis on their own. So they could, the idea was to teach them how to do the analysis, and they could do their own analysis step by step because they have the knowledge. I don't know about their specific studies, and I don't have any clue about the different species or what are expected there back then. And my idea was to empower them, that they can do their analysis on their own, and trying to find, to identify what are the best tools and approach where they could do their own analysis, but I could also do in an automatic way. So more the bioinformatics side aspect on running that on thousands of tools, samples in the same time. So then I decided somehow to develop some sort of environment that is called ASaiM, where the idea was to analyze gut microbiota shotgun sequences. With one aspect will be the expert database that combine information from intestinal microbiota that has been collected from different public databases. And another aspect will be the modular and user-friendly framework, where the people could process and analyze microbiota sequences. I will not really discuss more about the database now, because you will see later. But yeah, I will focus really on the framework aspect. And so the idea framework was really to develop a bioinformatics framework where we could generate workflows to analyze gut microbiota data. So we should be able to generate workflow with as many tools as we wanted. It has to be quite user-friendly, so that anyone, even that never used the command line, could use them. They need to have some flexibility and modularity, that we could choose the parameters that we want. We could choose the order of the tools, depending on the questions, depending on the samples or how it has been sequenced, etc. And we need to be able to incorporate in that framework the tools that the people in the lab wanted to really use. The one I could choose, me as a bioinformatician, so with the bioinformatic view, but also the one that my colleagues wanted to use because they heard about that. And also the databases. So it's quite some requirement for the framework. Back then, I tried several things, I must admit. I tried simple Python script. So I come from Python background, so my first things, okay, I will try with Python script. Okay, nice try. I will not do that again. The second aspect was to try to use workflow managers. So at this time, there were no SnakeMake, no Nextflow. So there were some workflow managers like Luigi from Spotify or Airflow from Airbnb, but they were not really developed for data analysis themselves. They were focusing mostly on the different the different steps and not on the flow of the data. So it doesn't really fit with the idea of bioinformatics analysis, where the flow is through the data, and what is important is the type of data that connect the different steps of a workflow. So then I started to try to develop something homemade approach. So back then I heard about Docker. So Docker, the idea of Docker is a containers where you could ship inside tools, all the dependencies of the tools that then get installed inside the Docker image. Then you don't have to so Docker is something where you take that, you install that on your computer, and everything that is inside you need there is inside installed. All the dependencies are managed, and you don't have issue with the versions of the tools, etc. So it's really great for reproducibility. Then with that, I could manage really the dependencies aspect and tools aspect. Then the idea was to then create the workflow. So what are the different steps that we need for the workflow, for the different for the analysis itself? So I created a simple web interface where the users could define which are the tools they want, in which orders, with which are the parameters, and it generate a JSON file, somehow a configuration file, and then they needed to run that with the command line. But we are back to the idea that they need to use the command line, which was a bit too cumbersome. And also, I mean, yeah, it's not really user-friendly, except the interface. So it was not really the best solution back then. Yeah, so ASaiM framework, I tried several things, was not really successful back then. So then I started to look and try to investigate if there are any things already available that could do the job somehow. What are my requirements and that. So you may have heard about Galaxy. So Galaxy was developed more than 15 years ago. It's an open source framework where it gives a web interface for thousands of bioinformatics tools. So command lines tools that we could use with the command lines, but that make available via a web interface where all the parameters that are exposed in the command lines are wrapped or available or rendered nicely looking inside Galaxy, inside the web interface. So if you look on Galaxy, so you have on the left here, you can see you have the different tools available. So what are the different tools you would like to use? On the right, you have what we call an history, where you can have the different, your data are available in your history. So you upload your data, you click on upload, and then you get your data that are available in your history. And then when you run a tools, so you click on the tools on the left, you choose your tools. And then if you open, for example, FastQC, you have the different options available for your command line that are exposed and nicely rendered inside in the middle of the interface. And then you could choose what are which inputs you want to have. So here it seems that I don't have any FastQ file in my history, but then it's filtered depending on the type of the format of the data or the requirement for these tools. And there is a different parameters, etc. And then once you click, we cannot see, but there is a execute buttons, then the output of all of these tools will appear in the history on the right. And it keeps tracks of everything, all the parameters that has been used, the version of the tool that has been used there. And it's also possible to interact programmatically with Galaxy. So if you are somehow allergic to the web interface, you could use a strong API to interact with that and never see this interface anymore. Another really nice, sorry, something nice with Galaxy, it has also a really powerful workflow builder and manager. So there is a way to build workflow within the Galaxy interface. So once you open the page for building workflows, you have all the tools available on the left, and then you can drag and drop their tools in the middle, and then you can connect the different steps or the different tools by clicking and dropping and connecting the different steps together based on the outputs and inputs of these tools. And if the formats are correct, then you can connect them. If the formats are not the correct one, so the output of one is, for example, FASTA and the input of the other tools is a FASTQ, then you will never be able to connect the two of them. So it's really helping to build a workflow, and then on the back, you can run this workflow automatically as much as you want on as many data as you want there. So it was a quick introduction to Galaxy. Back then, so 2015, in my mind, Galaxy was not really appealing, I would say. So I had really this, I don't know, bioinformatic feeling that, okay, Galaxy is not for me as a bioinformatician. So it's not only for the users. I don't see why I should spend time on that. But I was in a wet lab where most of the people here cannot use the command lines, and it was okay, I should give it a try at least. Yeah, I thought, okay, I give it a try. I will see if we can build something out of that. And by starting to look at it, I started to really dig into the Galaxy project and start to think, okay, it seems to fit more and more of the requirements that I had for this framework. So it's helping to generate workflow with as many tools as we want. It has this user-friendly interface. It has the flexibility of modularity, so we can choose, we can build the workflow as we want, either from the interface or by step applying the different tools that we are interested in. We can add as many tools as we want into Galaxy and the database that we are interested in. And it has really this aspect of transparency and reproducibility that is really important in science. So Galaxy keep a track of everything. So all the tools that has been, the version of the tools, the different parameters that has been used, etc., etc. It's really a core aspect of Galaxy. So I was, okay, more and more convinced that maybe Galaxy was the way to go there. So I was looking at the Galaxy interface, and there were many tools that were not really interesting for my users there. So I decided to create some sort of simple Galaxy instance with a custom configuration that really focused on the microbiome analysis, data analysis that are needed for my users and for building these databases with a standard workflow. So by then, I integrated a lot. So there are some tools available in Galaxy that were available to process our data, or the tools that my users wanted to use, but many tools were still not available. So I spent some time to integrate this tool into Galaxy. What does that mean, integrating a tools into Galaxy? Is to develop some sort of wrapper around the command line tools to export the different options, and that they look somehow nice on the web interface. It's mostly it's developing what the format there is XML. So it's quite interesting to see how developing Galaxy tools. And so I integrated many tools that my users wanted. So they were focusing on gut microbiota, and back then they were really interesting to get metaphylan and human tools from the Utonover lab into Galaxy. They were not available, so I spent really a lot of time and energy to import them into Galaxy and make them somehow helping the users to identify, to help them getting the correct parameter, the correct orders, etc. I developed also some workflows that could be used by the users. I also spent time on simplifying somehow the Galaxy interface, that the tools are ordered in a way that helps the users to know which tool they need to use for each of the steps when they do their analysis on their own step by step. So yeah, I ordered the tools. As I mentioned, I integrated some of the tools that were missing there. I also developed some workflow that were used standardly in the lab afterwards. So, for example, this row shotgun metagenomics data analysis with a first step of pre-processing. So at this time it was running FastQC and trim galore for doing the quality control and the trimming of the sequencings to improving the quality of the raw sequences. Then a dereplication phase steps to remove the duplicated sequences. A step of sort me RNA to sort the rRNA sequences and the non-rRNA sequencings there. From the unique reads, we then run steps of taxonomic assignation using metaphylan 2. And then we got an output as the taxonomic tables with the different taxon and the abundances that we could visualize with Graphland, Corona, and split the tables into a table for each of the different taxonomic levels. The non-rRNA reads were used as an input for human for doing the metabolomic assignations, and out of human we got the gene family abundances, UniRef at this time, that we could combine them in Go terms. And then we had the pathway abundances using metasys pathway there. And then we combined the metaphylan and human outputs to get information about which species is involved in which pathway or in which gene families. So it's an overview of the workflow that I developed for the team and that I could use afterwards for the databases. While developing that and integrating all the tools into Galaxy, I spent quite a lot of time also on documenting everything. So all the tools that are needed, but also how to use the interface, how to deploy that. So the documentation was from a user perspective, but also from someone that wants to deploy the same infrastructure somewhere else. And beyond that, so everything was stored on Github. So I created a Github organizations where I have a repository for the different tools that, for the different wrapper for Galaxy that I developed. And once the wrappers are developed, I push them on the Galaxy toolshed. So the Galaxy toolshed is like an app store for Galaxy tools. It's where you could find the different tools that are available and make available by the community that can be installed on any Galaxy servers. So when I developed a wrapper for my tools, I put them on the toolshed that anyone, anywhere could install these tools on their own server. And so I stored all these wrappers on the source of the wrappers on my GitHub repository. I created a GitHub repository also to really have with some shell scripts where to deploy this Galaxy server, this custom Galaxy server that get the latest version of Galaxy, that prepare the database and the tools and launch then afterwards Galaxy. So then I use that to deploy Galaxy and this ASaiM flavors on the local clusters. And I had the repository with the documentation I showed you. Just to mention that they were my first GitHub repository. So it was really the way I learned how to really use GitHub, which is an interesting, way that. So I was mostly alone working on that. So it was a step-by-step process by learning on my own. But then it became a bit bigger because so I started to contribute really to the Galaxy project. So from my own developing my own wrappers, I started also to get a bit more confident, and then I could contribute to existing Galaxy tools. I started to update some tools that were available and developed by someone else on a different GitHub repository. And I added some of my tools then that were not microbiome focused. I had them on a different GitHub repository. So I started to play this game of the open source and contributions. And I started to get also in touch with the community by participating to different community events. For example, the Galaxy Days organized by the French community. Or my first, it was really my very first hackathon where I participated to the contribution fest on developing metagenomics tools. So it was really nice. That was organized by the Galaxy community. And I met some people, and especially, so I met Björn Gröning. I will come back to him because, yeah, he played quite a role afterwards. So 2015-16, just to recap, so I developed tools. I started to really focus on Galaxy there, integrating tools that were mostly needed by the local users, and developed some workflows, developed the documentation, write the deployment for this custom server, make that available for the lab locally, and I was mostly focused on gut microbiota back then. Just to mention, yeah, my contract ended in the middle of 2016, and I didn't really have the time to work on the expert database back then. As I mentioned, so my contract ended, and I was not sure what to do. So then Björn, that I already mentioned, offered me a job there in Freiburg in Germany. So I moved to Freiburg, Germany. And then I started to work beyond the gut microbiota and beyond the local effort for my, my lab there. The first things is I started to really coordinate more the efforts for developing tools with the global Galaxy community, and especially, so I started to work with Saskia Hiltermann. She's amazing. And so we started to work really to develop more tools, integrate more tools into Galaxy for gut microbiota, for microbiota analysis. And we developed, we worked with the Intergalactic Unit Commission, which is a group of people interested in developing the best practices in tool development for Galaxy, but also integrating really great tools into Galaxy. So we put a lot of efforts all together to integrate the ASaiM and other tool suites into Galaxy. It was also the time where Galaxy decided to use Conda to manage all the dependencies. So Conda is a package manager, so it's open source package management that manage all the dependency, that help to manage all and install all the dependency on your own computer, but also on the cluster, etc. So 2016, there were a big effort of the Galaxy community to migrate all the tools and all the dependency to Conda. So we put a lot of efforts to migrate everything to BioConda back then. And 2016 is when we started with Saskia, big efforts on developing training materials with the Galaxy Training Network. So we developed an infrastructure that I will present the next slide to support training materials globally. So, for the Galaxy Training Network, we developed with the Galaxy Training Network online, a website online where you can find, now I think it's more than 200 tutorials to learn about different bioinformatics, but not only, using Galaxy. So there is different topics, for example, transcriptomics, etc. But for each of these different topics, you can find some introductory slide decks. So for example, because we were interested in the metagenomic aspect with Saskia, so we developed tutorials for introduction to slide for introduction to metagenomics, and we developed also some tutorials that are like for self-study or instructors. That are really step-by-step guides through a full analysis that mostly following scientific stories, so reproducing a paper or analyzing an interesting data sets. And by doing this step by step, we provide some scientific and technical backgrounds of the different steps that are needed. So why we need these steps, how we can do that, but also some hands-on sections. So some aspect where the person doing these trainings can really do that directly on Galaxy. So they got the data into Galaxy, then they can run those different steps that they can get an idea why they do that, see directly the result of the tools, select the different parameters, get an information about that. And because it's important, because learning by doing is really a really good way to get really familiar with especially with bioinformatics. So everything, the tools, the different workflow, the data sets, everything is available within this infrastructure. And in the back, it's one big Github repository that store everything and then serve everything, make that available at the website. You can find more information on training.galaxyproject.org, and we have a specific topics on metagenomics. So we developed some training materials, and then in the lab, where I was, they were not so interesting really in metagenomics per se, but one of my colleagues is working on CRISPR subtype discovery. And one thing he wanted to do is to try to identify new CRISPR subtype within metagenomics data sets. Within metagenomics data. And one thing for that, they needed longer sequencing than just the Illumina, the output of Illumina that you got. So then I started to work on and see what can we do for metagenomic assembly. So this project didn't really started as a Galaxy focused project. It was mostly a local project, and my idea was to use the local cluster for that. But I realized that, okay, metagenomic assembly, you need quite a lot of resources, CPU memories, and it's not so easy to know how much memories or how much resources you will need before running the assembly itself. So because I could use the Galaxy server that we are maintaining in Freiburg, and then if I could run that within Galaxy, I don't need to care about the resources. I'm not the Galaxy admin there. I could just submit my jobs to Galaxy, and everything is done on the back, and I don't have to think about submitting my jobs to the cluster. It's done automatically by Galaxy. And I thought also, okay, if I need these assembly tools, maybe someone else needs them. So then I spent some time to integrate some metagenomics assembly tools, so Mega8, Metaspace, and also some assembly quality tools like MetaQuast, Valet, and other tools. And then I built a workflow for metagenomics assembly based on Metaspace and Mega8, because they were the state of the art at this time tools. And then I could run them automatically on many thousands of data sets. And then my collaborators or my colleagues run then their tools, their CRISPR subtypes discovery tools on the outputs. 2016 also put a lot of efforts to, 17 mostly, to put a lot of efforts to make ASaiM more redistributable and shareable with others. So as I said, so it's built really on Galaxy. It's used all the Galaxy aspects, so the Galaxy toolshed to get the tools from, the BioBlend and Ephemeris that are two Python libraries to help configure and the databases, the workflows, etc. Conda to install all the tools and dependencies, and I put everything into a Docker images. So Galaxy and everything were inside a Docker image. And as I already mentioned quickly, but all the tools and workflow were then deployed on the Freiburg Galaxy server that we are maintaining in Freiburg, that make that available to more users. So to summarize, to 2016-17, we put a lot of efforts to integrate more tools for supporting more microbiota data analysis, so mother, SHAM, and assembly tools. We developed more workflows, a lot of efforts on trainings, coordinations with the community itself, so and make that available for more users via the Freiburg Galaxy server. Then came the time of spreading these resources to more people and supporting even more type of microbiome analysis. So the first step was to publish a paper on ASaiM that were published in Giga Science 2018. But the more important things is that, so at this time, 2018, beginning of 2018, the Freiburg Galaxy server became the European Galaxy server and was become open to everyone. So it's free for registration for everyone everywhere. And we got also, it's maintained, so this European Galaxy server is mostly maintained by European team that is mostly leaded here in Freiburg. And on this European Galaxy server, we have this different dedicated interface for the different type of analysis. So we have one now, microbiome.usegalaxy.eu, that is really focusing on metagenomics and microbiome data analysis, where you can have a subset of the different tools, because on the European Galaxy server, we have thousands of tools installed. So it can be tricky to find which tools you need to use for your own analysis. So with these dedicated resources, we have only a subset of the tools available. And you have a different interface when you join for the first time the Galaxy server. You can find interesting resources for your type of analysis you are interested in. Then we also worked with the Galaxy P communities. That is for they are more interested in proteomics, but then they were interested in the metatranscriptomic aspect. And so we worked together with the Galaxy P to develop, to modify a bit our workflow that we had before to really make it running for metatranscriptomic data. We developed a really quite comprehensive training for that really explain well why are the different steps, what we are doing that, and really from the science on the really focus, really a lot of information on scientific and technical aspect. We published that in F1000. There are also big efforts from the community, especially led by Saskia, to support nanopore data. So there is this NanoGalaxy where they integrated a lot of tools for nanopore data. So you have the QC tools for like pore shop, filtering, nanopolish, and all the pore tools data. Some tools for genome assembly, but also some specific for taxonomy tools. And Christo designed lately tutorials for 16 years microbial analysis with nanopore data that somehow follow this workflow there, with first steps of adapter removal with pore shop, filtering with FastP, quality control with FastQC and multi-QC, and taxonomic assignation with Kraken2 and visualization with Krona, which is a quite simple workflow, but still working well and with a big documentation in the back to help explain why we do these steps and how we can interpret the output of that. So with these tutorials, with workflow, we could also make that available to even a broader audience. So we started with the community in Freiburg, with some scientists that were interested to reach a broader audience. We started the street science community with a focus on reaching pupils, but also citizens, to teach them about science. And we focused at the beginning on the project that is called Decoded, where the idea is to get them immersed somehow in biology, bioinformatics, and science via the beer and the yeast. So the idea is we take a bottle of beer with them. So we are with them, we go to a lab, a wet lab. We take a bottle of beer, I think it's mostly 2 bottles of beers. We, from the beer, we extract the yeast, and then we extract the DNA of the yeast. So they do it, we don't do it. They do it, we explain them step by step how to do that. And then we have a minion with which we can do the sequencing. So then we got the sequences, and then we do the data analysis where we can we identify which are the yeasts that we sequenced. So it's really an introduction to the biology and the genomics for a broad audience. So everything is, we worked a lot to make the different protocols really as simple as possible. And we organized several workshops with that, so with pupils, with students, and with also citizens. For the different steps, so the extractions, we developed really simple protocols for the extraction and the sequencing. We put them available on the Street Science community website, and we explain everything, including the different steps. So why we are doing these different steps, what is it important, what can they see, etc., etc. For the data analysis, we use mostly the resources that has been developed by the NanoGalaxy community to create a workflow, a simple workflow that the people participating to the workflow can run, and then they could see the output as a corona visualization where they can really see the different yeasts and their abundances. And it was quite exciting. So we got nice results, and the people were quite excited afterwards. So 2018-19, yeah, opening even more spreading these resources to more with the European Galaxy server, with the publication, more tutorials, connection with different other Galaxy communities, and the outreach with the Decoded project. 2020, I was in parental leave, so I didn't work on ASaiM at all during this year. So it was a bit in pause there. And so 2021, I'm back. So then what else? Even expanding even more. So the idea now is to really start to build really a global Galaxy microbiome community. The first step of that would be to update ASaiM, so updating all the existing tools and workflow that we have with the new versions of the tools, because there were some changes over the last year. So human, we just finished to update human metaphylan. So SHIME2, it's ongoing. So there is a big work of the SHIME2 contributors of the team to integrate the SHIME2 into Galaxy, and we are working with them to do that. We will like to implement and benchmark more external workflows, for example, the Magnify and BioBakery. So I will go just quickly on the BioBakery workflow. So the BioBakery workflow are developed mostly by the Utenhover lab in US and also the Sigata lab in Italy. And the idea, they have different tools for different workflow from metagenomics, 16S, metagenomic and metatranscriptomic, to combine metagenomic and metatranscriptomics. And some of the tools are already available in Galaxy, and we would like to make them this workflow also running and making sure that they can run similarly on Galaxy also. The same for the Magnify workflow. So Magnify is the EBI metagenomics. Also, it's the same groups. And EBI metagenomic Magnify, it's a big database of a lot of it's really interesting resources with many data sets on microbiota, and they run automatically a lot of their workflow also on all the data set there, so we can really look at their data. And it would be awesome if we could manage to run that also similarly on Galaxy. So we will work also to integrate, to make sure that all the tools are available there, and we can have similar workflow there. Available for the users if they want to run that locally on their own. We will integrate even more tools and data sources and databases in Galaxy. So I started a big wish list of tools for Galaxy. So here is just a spreadsheet with many tools available. And every time I see a nice tools published or list mentioned somewhere in a publication, I usually put that here and I check if it's already available in Galaxy. So if it's available, if it's not, then it's become red. If it's available in Galaxy but not in the latest version, it's in yellow, orange there. And if it's green, it's the most up-to-date version. So we have already some tools there. Some are updated, but there is still work that we can do to support even more, make that tools available that the users can choose really which tool they want, because they heard that this tool is the best, then they can test that and they can even check and compare the outputs with other tools. It's really our aim that the users could choose, and then we will make an effort on that. And developed a lot training materials that helps also the users to choose the tools and understand what they are doing. So the idea would be to develop more training materials, mostly documenting the different workflow that I mentioned before, but also some sort of curriculum where you can from 0 to hero in microbiome data analysis. So you know nothing, then you go through the Galaxy introduction, and step by step you will become more and more experts into using Galaxy, but also how to run your own microbiome analysis. And with the aim that at the end we have the possibility to run Jupyter notebooks and RStudio directly within Galaxy. So we will also provide trainings for visualization or advanced visualizations of microbiome data analysis with tutorials on that, using the resources and RStudio or Jupyter within Galaxy. Then you could stay in Galaxy, and the users could just do that, and you could do everything within Galaxy. Then you don't need to move your data back and forth, losing track of what has been done. You have really a full track of everything, of the different steps. So for producibility aspect, it's really great then. And so we will work with the Gallantries. So the Gallantries is a European funding project for which we got the funding starting last year for 3 years. And the idea is to develop more training curriculum on data management, data analysis, etc. And one track will be really focusing on metagenomics, microbiome data analysis. But more globally, we will kick off really soon the global Galaxy microbiome community, with the idea to organize regularly meetings, to have a mailing list, chat directory, and whoever is interesting, to coordinate really our efforts on tool developments, new database integrations, and training development. And also, if we need we have some specific needs that need to be changed on the Galaxy interface, on the Galaxy framework itself, then we can centralize these needs. And really, if we are several people that need that, we can really ask there that needs to be developed, or needs us to be something important that the core Galaxy team need to work on soon. The idea is also to centralize the communications effort with other communities or groups. So within the Galaxy community, so there is different working groups, different communities. There is this steering committee that oversee everything there. So if we could get a way to have a centralized way to know what's going on in the different communities, but also outside Galaxy. For example, the Microbiome Info Slack. I don't know if you know about that, but it's just amazing. I can just recommend everyone to go there. You will find every day a lot of new stuff and amazing stuff. You learn a lot of amazing stuff there. So it would be nice. There is in this Galaxy Microbiome Info Slack, there is a dedicated channel for Galaxy. So it's nice if we could coordinate also more the efforts together there. And share the interesting resources on which we need to keep an eye. For example, the nice workflow that will be nice to have into Galaxy, or nice projects that are going on for which we could help them to get the stuff also in Galaxy. If you are interested to join that efforts, here are the links to a spreadsheet where you can add your name there. And we will kick off really the meeting really soon, probably during the next Galaxy Community Conference that will happen beginning of July. And so if you want to join that, I can recommend you to just add your name there, and I will send an email really soon for that. So a global overview, 2021, it's just the start. Okay, we are already end of May or beginning of June, but there is many things that will probably happen this year. So I really work a lot on microbiome, and I know that there is a big effort also globally to work on microbiome analysis. And so we will do a lot of work on tool updates, new tools supporting more workflows, developing this Galaxy for Microbiome community. The ASaiM MT paper that I mentioned was just published this year. And I didn't mention it, but for the decoded project, because currently we cannot do the workshops because of the pandemic, we decided that we will develop some sort of online game to get the people learning about the microbiome analysis, about microbiome in general, but also learning quickly how to do a microbiome data analysis from the decoded data that we will get. So we are currently working here in Freiburg on developing this game that will be mostly based on Galaxy, so using already existing resource structure of Galaxy. And we have a student that will start soon really implementing that. We have the story, we have everything. We now need to get it on Galaxy, and hopefully by the end of the year, we have already a prototype of that. So I hope with that presentation, I'll show you really how ASaiM started from a local project for my lab to really starting to grow and starting to become a global effort to support microbiome data analysis in the large sense, and to really support everyone that is interested to do microbiome data analysis. And for that, I would like to thank many, I probably forgot many people and many and logos and stuff, but first I would like to thank the French Conseil Régional d'Auvergne, which were the fund and the University of Auvergne that funded me at the beginning of the project. When I moved to Freiburg, and since then I'm funded by DENBI, which is the German network for bioinformatics, and I'm part of the Backofen lab and the Freiburg Galaxy team. I would like to thank the European Galaxy server because it's where we can run everything nowadays. The Galaxy project in general, because without that, it will not be able to do that. The Galaxy training network for making the training materials and supporting that community. So we have currently 170 contributors. It's just amazing. Elixir for the European bioinformatic infrastructure somehow, the street science, and many people there. I put some people there, but there is many more people beyond that. So I would like to thank, and I would like to thank you all to listening to this talk.